Okay, in this video we're going to talk about the different types of bone development. So ossification, also called osteogenesis, is the process of bone tissue formation. And formation of the bony skeleton, it begins within the second month of fetal development. And we talked about how most bones in your body start out as cartilage, so that basically ossification occurs in month 2, but it's basically osteogenesis of cartilage. Now, postnatal bone growth occurs until early adulthood. So we're saying that after birth, bones continue to grow, and we'll talk about and differentiate between those different types of bone growth. And bone remodeling and repair are considered lifelong as well. Now, in terms of formation of the bony skeleton, there are 2 major forms of ossification or bone development. Up until about week 8, the fibrous membranes and hyaline cartilage are replaced with bony tissue. Through 2 major mechanisms, we have endochondral ossification and intramembranous ossification. Endo means within, chondro is cartilage, so endochondral ossification is actually bone formation within a hyaline cartilage model, so that bones that are endochondral bones, basically cartilage, they form most of the skeleton. So most of the bones in your body start out as cartilage, but then are ossified through endochondral ossification. Now, intramembranous ossification is actually where bone develops in fibrous sheets of membrane, typically forming flat bones. So endochondral ossification is the first type we'll talk about here, and it forms essentially all bones inferior to the base of the skull, except for your clavicles. This begins late in the second month of development, and it uses previously formed hyaline cartilage to serve as the model for bone tissue formation. And it requires the breakdown of that hyaline cartilage prior to ossification, so that that cartilage is basically ossified into bone. And it begins at what we call the primary ossification center. Now you find this in the shaft of the cartilage, and essentially blood vessels infiltrate the perichondrium, converting it to periosteum, and then mesenchymal cells specialize into osteoblasts, which then can turn into that particular type of connective tissue, which is bone. Now, the 5 major steps of ossification are bone collar formation. This is the first step that it begins around the diaphysis of the cartilage model. The second step here is that cartilage in the diaphysis then calcifies and develops cavities. Periosteal bud then invades the cavities, leading to formation of spongy bone. And the bud is made of blood vessels, nerves, and red marrow, as well as different types of cells like osteoclasts and osteogenic cells. The fourth step here is the diaphysis elongates and the medullary cavity forms because of the breakdown of that cartilage. And the secondary ossification center also appears in the epiphyses. The fifth step here is that the epiphyses ossify, and then hyaline cartilage remains in the epiphyseal plates, where it's sandwiched between the primary and secondary ossification centers. So what this slide is showing is basically the process of endochondral ossification, and it gives you 1, 2, 3, 4, 5 steps, as well as the timeline here. We see that up until about the 3rd month, most of your bones are still cartilage here, and you see that the first step of bone formation is the formation of this bony collar near the diaphysis of this hyaline cartilage model, and then we get the formation of a primary ossification center here in the center of this diaphysis. Now, in the second step here, we get deteriorating cartilaginous matrix, where the primary ossification center begins to expand the bone outward and upward, and the bony collar also thickens and elongates. The third step here is the periosteal bud invades the primary ossification center. This brings in your blood vessels, your nerve fibers, and osteogenic cells and osteoclasts, which effectively then turns this area into technically bone, because now it's got blood vessels. And then the fourth step here is that the diaphysis begins to elongate. All the cartilage is actually eliminated from the diaphysis, so what you're left with is just bone. And then the secondary ossification centers appear right around the time of birth. So we're saying that at birth, there really aren't much of an epiphysis at this point, right? So the secondary ossification centers in the epiphyses have just started to form when someone's born, which kind of makes sense because you want their bones to become flexible and movable in order to be more easily passed the birth canal. What we find then is that from birth up until childhood and adolescence, we see that the secondary ossification centers continue to grow, expand, until essentially what you're left with is what looks like a long bone. So you have your epiphyses at the ends here. We have our diaphysis. And the only cartilage that's left from the hyaline cartilage that it began as is essentially the articular cartilage that you find at the ends of the long bones, as well as the epiphyseal plate. Now, the epiphyseal plate is basically this little band of cartilage trapped between the secondary and primary ossification centers. And it's essentially a band of cartilage that just didn't fully ossify, and this is the growth plate. So this is going to continue to divide and elongate the bone through childhood and adolescence. In adult, this epiphyseal plate is fused, and what you're left with is an epiphyseal line, which is just a little line of compact bone. Now, this differs from intramembranous ossification because intramembranous ossification is a more rare type of bone formation, but it effectively forms most of your cranial bones and flat bones. So like your frontal, parietal, occipital, temporal, and clavicles are all formed by intramembranous ossification, and it basically occurs within sheets of mesenchymal fibrous tissue. So there are 4 major steps involved here. We have ossification centers that are formed when these mesenchymal cells cluster and become osteoblasts. These osteoblasts secrete osteoid, which is then calcified. Woven bone is formed when osteoid is laid down around the blood vessels, which forms a trabecular appearance of spongy bone. And then the lamellar bone replaces woven bone, and what we find then is that red marrow appears within the woven bone spaces. So in terms of these steps, you have 1, 2, 3, 4. And the first step here is that these mesenchymal stem cells within the fibrous tissue can start to aggregate and form clusters. These clusters then can differentiate into osteoblasts, which then start to lay down osteoid. Remember, osteoid is the unmineralized organic bony matrix, which is basically calcium-binding protein and collagen fibers. Now, after the osteoid is laid down, effectively that gets mineralized by hydroxyapatite, which are calcium phosphate salts. Some of these cells get trapped in the osteoid itself, in the mineralized osteoid. And this is actually a lacuna now. So you see that this cell that's trapped in the lacuna, well, it's now called an osteocyte because it has no choice but to become less active, more dormant, because it's now trapped within a space in the bone. Now we get a line of osteoblasts at the outer margin that continue to lay down new osteoid until effectively what you form is woven bone. Woven bone is basically, it looks like spongy bone, but it's basically just bony matrix that is wrapped around the blood vessels that traverse through this connective tissue. Now, essentially the 4th step here is that osteoblasts continue to line up until they lay down lamellar bone on the outside edge, basically like a bony collar that surrounds the outside edges. This will later become what we know as compact bone, with our spongy bone here trapped in the middle. Now in the 4th step, also what happens is the spongy material here, diplo, basically it's filled with yellow marrow, and that is a source of hematopoietic stem cells to help regenerate lost blood cells. So you can see that the process of intramembranous ossification is similar but different than endochondral. For one, it begins in sheets of mesenchymal stem cell tissue, and 2, it basically has one ossification center that essentially expands outward and eventually becomes our compact with spongy bone in the middle. Now, what we're going to do next is actually differentiate different types of bone growth after birth. So the first one we'll talk about here is interstitial bone growth. So interstitial bone growth is a lengthening of bone along the longitudinal length of that bone. So this is actually a type of bone growth where bones can increase in thickness and in length. And bones stop growing during adolescence because of the loss of the epiphyseal plate, but some facial bones continue to grow slowly throughout life. So interstitial bone growth requires the presence of epiphyseal cartilage. Remember, epiphyseal cartilage is what you found in long bones, and it's basically a little band of hyaline cartilage that's trapped between the epiphysis and the diaphysis, and it's essentially that hyaline cartilage that's left behind from development. Now, the epiphyseal plate maintains a constant thickness, but the rate of cartilage growth is balanced by bone replacement. So we find that this epiphyseal cartilage divides. It continues to make new cartilage. But this new cartilage is essentially turned into or ossified into new bone. And what we're saying then is that cartilage will grow, die, and then get converted to new bone tissue. So we can divide our epiphyseal plate into 5 zones. We got resting zone, proliferating zone, where the cartilage is dividing, We have the hypertrophic zone where these cells get larger and begin to die. We have the calcification zone where the dead cartilage gets calcified. And then we have the ossification zone, also called the osteogenic zone, where that dead calcified cartilage is then converted to bony tissue. Now, the resting zone here is an area of cartilage on the epiphyseal side of the plate that's relatively inactive. And nearby we have the proliferating zone. That's on the diaphysis side of the epiphyseal plate, and it consists of chondroblasts that are rapidly dividing. These actually help to create new, extracellular material, and essentially making new cartilage, which pushes the epiphysis away from the diaphysis, causing lengthening. Now, the next zone here is where the older chondrocytes closer to the diaphysis start to enlarge and erode, forming these interconnected spaces. And by the calcification zone, it's basically dead cartilage. So the chondrocytes die and deteriorate, and in this zone, we have basically calcified cartilage that's dead. And in the fifth zone, that calcified dead cartilage is essentially turned into bony tissue. So we're seeing here is that chondrocytes deteriorate, leaving these spicules and the calcified cartilage at the epiphyseal diaphysis junction. These spicules are eroded by osteoclasts and then covered with new bone tissue by osteoblasts, and ultimately replaced with spongy bone. The medullary cavity will enlarge as the spicules are eroded. So we're seeing here are those 5 zones of the epiphyseal plate. So this should have an epiphyseal plate here. And if you zoomed in on this, you'd find we have a resting zone, then our proliferating zone where the cartilage is dividing, our hypertrophic zone where the cartilage cells get larger, our calcification zone where the chondrocytes die, and then you're left with calcified dead cartilage. And then we have the ossification zone where spicules are eroded by osteoclasts and then converted to basically osseous or bony tissue by nearby osteoblasts. You can imagine then that this is going to divide and push downward, and as it gets pushed downward, the cartilage is dying and be converted to bone. This is effectively what helps bones grow in length. Now, in terms of what determines the growth of bones in length is that near adolescence, chondroblasts divide less often. The epiphyseal plate becomes thinner and ultimately is replaced by bone completely. We call this epiphyseal plate closure when the epiphysis and the diaphysis fuse together and there's no more epiphyseal cartilage. Now we have the epiphyseal line, which is just bony tissue there. Bone lengthening ceases in females around age 18 and in males around age 21. Now, in terms of bone growth in width, we call this appositional bone growth. And this is where they widen as they lengthen as well. Bones become thicker in response to increased stress, and osteoblasts beneath the periosteum secrete bony matrix on external bone. Osteoclasts remove bone from the endosteal surface, and what we see then is that actually, as a consequence, a widening of bone. Because osteoblasts are laying down bone on the outside, osteoclasts are breaking it down from the inside, ultimately causing the whole bone to become wider, but proportionally the same thickness in terms of compact bone. Now, there's usually more building up than breaking down, which leads to like thicker, stronger bone that's not too heavy. So what we're seeing here then is actually bone growth, and we're seeing that the bone can grow in length. And what we need then is also bone remodeling. And bone remodeling includes appositional bone growth. Appositional bone growth is actually where bones widen as they lengthen. You can see for one, yes, the bone lengthened because it's slightly longer than it was in the beginning. That's because of this epiphyseal cartilage dividing and then basically laying down new bony material on the diaphysis side. Now we also see a corresponding widening of the bone because of appositional bone growth, where essentially osteoblasts lay down new bony matrix on the outside, osteoclasts break it down from the inside. That's what you're seeing. This dotted line means basically where the bone used to be. You can see that all this bony material was broken down, but then it was expanded outward here by osteoblasts. So what you end up with then is a longer bone because of the endochondral, the interstitial bone growth here, and a wider bone because of the appositional bone growth. Now, there's also lots of hormones that can regulate bone growth, and we see that growth hormone is one of the most important hormones in stimulating epiphyseal plate activity. Thyroid hormone plays a role as well as testosterone and estrogen in males and females. And excesses or deficits in any hormone can lead to abnormal skeletal growth. Like if someone has growth hormone excess, that can lead to gigantism, Or if they have growth hormone deficiency, it can lead to lead to dwarfism, where they're actually abnormally short.